Hello, everyone. I would like to thank the organizing committee for inviting me to present this work. I'm basically a physiologist. I'm specialized in neuroendocrine physiology. I have interest in neurophysiology. And this work was published in Behavioral Brain Research last March. This work title is Vitamin D3 Improves Decline in Cognitive Function and Cholinergic Transmission in Prefrontal Cortex of Streptozotocin-Induced Diabetic Rats. Diabetes mellitus is a very important disorder. It's very common all over the world, and it is one of the most costly diseases. Complications of diabetes mellitus are diverse metabolic disorders, cardiovascular problems, renal, endocrine deficits, besides two neurological disorders. Neurological complications of diabetes mellitus have included learning and memory deficits besides to many others. Research have shown that both type 1 and type 2 diabetic patients have or suffered from reduced performance in cognitive domains, and they performed worse than control subjects in memory and information processing tests. Also, when diabetes is induced in experimental animals, many works have recorded potential changes in cognitive functions, and diabetes has been considered as a risk factor for dementia. Regarding the possible pathophysiological mechanisms that could underlie the brain dysfunction, diabetes mellitus, many have been searched and proposed, like microvascular alterations in the cerebral circulation, oxidative stress and overproduction of reactive oxygen species, accumulation of amyloid beta, apoptosis and impairment of neuronal survival, in addition to disturbances in neurotransmitter balance. Acetylcholine is one of the neurotransmitters that are important for integrating functions of learning and memory. As we can see, acetylcholine or cholinergic fibers, they project in the basal forebrain and originate in the basal forebrain and project to many areas in the cerebral cortex. And regarding the prefrontal cortex, they project to many layers, and they are important and for the memory functions, the novelty-induced arousal, the encoding of novel stimuli, and the memory consolidation. Depletion of acetylcholine was proved to participate or play a role in the memory deficit of Alzheimer's disease. But concerning diabetes mellitus, the cholinergic status have not been much investigated or confirmed, and neither or nor its possible relation to any cognitive impairment that could happen in diabetes mellitus. Regarding management of such memory deficits in diabetic patients, trials have shown that anti-diabetic drugs, they cannot do things for this memory or learning deficits or impairments. While vitamin D has been emerged throughout the last 10 or 15 years, and large community-based studies have proved the association between vitamin D deficiency and many neurological disorders, including Alzheimer, Parkinson, epilepsy, and some psychological problems, depression, schizophrenia. All these are associated to vitamin D deficiency. Furthermore, the active form of vitamin D, when supplemented, it showed neuroprotective effects, including anti-apoptotic, anti-inflammatory, anti-oxidative. And that's why this study or this work aimed to examine the possible beneficial effects of vitamin D3 supplementation on the episodic memory as a measure to assess the cognitive function in streptozotocin-induced diabetes, and to investigate the alterations and markers for cholinergic transmission in the prefrontal cortex as a possible underlying mechanism. In this work, young male Western rats were used, and we tried to choose this age to avoid any possible age-related alterations in the cholinergic transmission or impairments in memory. They were made diabetic by injection of streptozotocin, 45 milligram per kilogram body weight. And diabetic animals were classified into diabetic group without any treatment, and diabetic group which was treated with 500 international units per kilogram per day through oral route, of course, in addition to the control group. The novel object recognition test. At the end of the 10 weeks, all rats were subjected to the novel object recognition test. This is a highly validated test to assess memory in rodents. And that test does not require training, lengthy training, and in addition, it does not induce a lot of stress for the animal. As we can see in the beginning, we put the animal in the first cage, which is called the trial cage, with 2 identical objects for 3 minutes. Then we transfer the rat to the home cage. Again, we return it to the test cage with 1 familiar object and 1 novel object. Rats, they have innate preference to explore the novel objects. So if it spends more time exploring the novel object, this means good memory function. But if there is indifference between exploration of both familiar and novel objects, this means that there is memory deficit. In our experiment, the rat spent 3 minutes in the training session with the 2 familiar objects, then transferred to the home cage for nearly 15 minutes. Again, we transferred it to the test session, 1 familiar and 1 novel object for 3 minutes. This 15 minutes, all researchers who had worked on that test, they plotted like forgetting curves, which gave the appropriate time for working of short-term memory, and this the 15 minutes is nearly that proper time. The total exploration time of both in the trial and excuse me, exploration of both, it's not working anymore. Exploration of both novel and familiar objects in the trial and test sessions was calculated, besides to the discrimination index. The discrimination index equals the exploration of novel minus exploration of the familiar divided by exploration of both novel and familiar. This difference is presented like a percentage of the total time spent in exploration. Then all animals were sacrificed, the prefrontal tissue was homogenized and centrifuged to obtain the supernatant. This supernatant was used to assess for 2 important enzymes, the choline acetyltransferase, which catalyzes the formation or synthesis of acetylcholine, and acetylcholine esterase, which catalyzes breakdown of acetylcholine. As we can see, the choline acetyltransferase, which catalyzes formation or synthesis of acetylcholine, it was significantly decreased in the diabetic group. It seems it's running out of battery. No problem. Okay, it was significantly decreased in the diabetic group. When diabetic rats were treated with vitamin D, there was again improvement in the activity of choline acetyltransferase when treated with vitamin D. On the reverse, the acetylcholine esterase enzyme, which breaks down acetylcholine, was markedly increased in the diabetic group when compared to the control group. And again, when the diabetic rats were treated with vitamin D, the activity of this enzyme was significantly decreased, still not to the control level, but significant when compared to the diabetic group. This is the total exploration time in both the trial and test sessions, and it's indifferent. There is no significant difference between the 3 groups. While the discrimination index was significantly decreased in the diabetic group, they spent less time exploring the novel object. And when they were treated with vitamin D, this showed significant improvement in the exploration or improvement in the memory deficits or in the memory conditions, so they explored or spent more time exploring the novel object. In conclusion, this work confirmed the cognitive deterioration in diabetes mellitus, postulated important mechanism, which is a decline in cholinergic transmission in the prefrontal cortex, and gave an evidence for the possible beneficial effect of vitamin D3 in protecting the cognitive function in diabetic rats, and highlights the importance of vitamin D supplementation to diabetic patients. Thank you so much, and thanks goes to my colleague, Prof. Abdelmanan. Thank you.
Any questions? Yes.
Is there any idea of how, if it's only D3 or will other vitamin D subtypes also help?
Vitamin D, the well-known types of vitamin D, vitamin D2, which is plant in origin, and vitamin D3, which is formed photochemically in the epidermis, in the animal epidermis. This is the form which can be synthesized and that we usually take it as a supplements. The active form of vitamin D3. Almost all the researches concerning vitamin D are regarding the active form of vitamin D3, 1,25-dihydroxycholecalciferol. No, I'm so sorry. Most welcome. Okay, no, sir.
Anyone else? Yes.
Very good work. That's excellent.
Thank you.
With rats and looking at cognitive, assessment is the most tedious thing you could do. It's not an easy thing. How many animals did you use?
I used 10 animals in each group. I used, at the end, we started with a bigger number, around 12, but at the end I got, nothing was passed out, but we got, for the statistical evaluation, 10 rats for each group.
So 10 is, you didn't see a huge degree of variability. It looks like 10 is too many. You did that in Saudi Arabia? The project, what did you do?
Yes.
Do you have, in our university we have to go through an animal committee, animal approval.
Ethical committee, yes, we should got approval from the ethical committee.
If I use, like, say, for example, if I could achieve the results with 4 animals, they wouldn't allow me to use 10. And you didn't have a huge degree of variability. You could have got the same results with 5 instead of 10, right? Or am I? Or you didn't want to take a chance?
5, I think 5. We never worked with such, usually. Usually we used to, this 10 could give acceptable, but lower than this, no, we never.
And he used that was the vitamin D, 125-dihydroxycholecalciferol.
The active form.
What dose is that?
The dose? 500 international unit. There are many studies regarding the dose, and that dose gives our makes the level sufficient, prevents. We cannot give bigger doses because that might cause toxicity, vitamin D and calcifications. So this is the most appropriate dose.
Do they have high incidence of vitamin D deficiency? Like, for example, Libya, all my 3 sisters, they have vitamin D deficiency. Is this the high incidence there in that area?
Basically, I'm Egyptian and I'm working for Saudi. I have not been there for so long time, but when I read about this, yes, it has become very common, vitamin D deficiency. I got interest in vitamin D deficiency when myself, I started to suffer like some muscle pains a few years ago, and I was advised to check my vitamin D level, and I found it around 12 nanomole per liter, severely deficient. So most of us, we can suffer severe vitamin D deficiency, and we are unaware. Even in the most temperate regions of the world, we do not usually expose enough to sunlight. That's why. But I think it's wide common in Saudi Arabia.
Did you look at long-term memory?
No, I didn't check long-term memory. I listened to your presentation yesterday, and you seem very expert in memory assessment in rats, and I remember the maze, multi-hole maze. But for me, I didn't check I checked only the novel object recognition test
Sure.
as a short-term memory.
Okay. Thank you very much.
Thank you. Thank you.
Thank you.